Welcome to another lecture series on amino acid MCQs. We'll continue our lecture series on question series on chemistry properties, reaction, classification, of peptides, and tests to test for the amino acids in solution, the proteins. So we'll continue our question. So the question 22. Amino acid buffer solutions because they are amphoteric. They have both they can both accept and donate the proton to the amphoteric. We have Ninhydrin gives the color with most amino acid purple color, but with amino acids like proline to give a yellow color. With proline, Ninhydrin gives a yellow color. The Xanthoproteic test is for aromatic amino acids. Thyroxine, phenylalanine, tryptophan, aromatic amino acid. The Millon's test is for aromatic amino acid that has a phenol, that has a hydroxyl group, like a phenolic group reaction. What do we mean by a phenolic group? If you have an amino acid, is NH3, is hydrogen, is CH2, then this group is a phenyl group. So this is a phenylalanine. Now, if the phenylalanine has a hydroxyl group, it is thyroxine. And this thyroxine will test positive to Millon's test. So this is the phenol. This is the phenol we are looking at for. So, but it can give a false positive test if there is presence of phenol in the solution. There is presence of phenol in the solution to give a false positive, because it's testing for phenol. Now, the Sakaguchi test is for the guanidino group of arginine. The guanidino group looks like this: N, C, N, N. Why this one goes back to your carboxylic acid. So this one goes back to, so this group here is the guanidino group, which is found in what? In arginine. It is found in arginine. You have the lead acid test, which is for sulfur-containing amino acid. Sulfur-containing amino acid gives a black precipitate with sulfur. So we have electrophoresis separate amino acid based on dash, pH, and charge. But for proteins, it is charge and size. But for amino acid, it's pH and charge. For proteins, it is charge and size. So paper chromatograph separate based on partition coefficient. What is partition coefficient? It is a separation that is based on the solvent partition. For example, if we have a paper immersed in a fluid or in a solvent, and our sample is docked somewhere here, and this is the solution. Let's say this is solvent. Let's say hexane. And what we have here is soluble in hexane. which is a non-polar solute, non-polar molecule. So as hexane migrates via capillary action, because of this molecule is soluble in hexane, it will push the molecule to come up here. It will push the molecule to come up here. But if the molecule is not soluble in hexane, let's say we have another one that is not soluble in hexane, let's say a polar molecule, it will not push the molecule. Then you now see that it is based on the solvent partition. So the molecule that wants to be separated must be able to dissolve in that particular solvent of choice. So aromatic acids are the building blocks of proteins. They are the building blocks of proteins. So biological function of amino acid includes enzyme formation of protein formation. Tryptophan is a precursor of serotonin. Serotonin is 5-hydroxytryptamine. 5-hydroxytryptamine, which is serotonin. So tryptophan is a precursor of serotonin. Thyroxine is a precursor of thyroxine, which is produced by the thyroid gland. We also have dopamine. So it's a precursor of both thyroxine and dopamine. Dopamine is a neurotransmitter. Histidine acts as a buffer due to what? The imidazole ring, which has a pK near physiological pH. So the physiological pH is around 7 point something, 7.2 to 7.4. Usually some tests will take it from 6.8, 7.2 or 7.4 as physiological pH. So histidine has a pK that is near that, though the histidine has three pK regions: the carboxyl group, the amino group. The carboxyl group is around 2 point something, while the amino group is around 9 point something, while the one on the arrow group is around physiological pH. So that's why I say it can actually serve as a blood buffer. Most amino acids occur in protein as dash. L form, they occur in L form, not D form. They occur in L form. Usually the carbohydrate occurs in the D form, while the amino acid occurs in the L form. And it is one of the properties of the peptidoglycan cell wall that makes the cell wall of the bacteria not susceptible to human enzyme, like the enzymes formed in the lysosomes, because the amino acid in the peptidoglycan wall is in the D form, and our enzyme cannot recognize the D form of amino acid. They only recognize L form of amino acids. So the answer is L form. So reactions of amino acid with nitrous acid produce alcohol. Nitrous acid produces alcohol. Now, it's actually a deamination reaction, but in the biological system, deamination reaction will produce a ketone. Decarboxylation produces an amine. So, if we have the amino group, O minus NH3 plus hydrogen, and let's say CH3, for example. If we decarboxylate the carbonyl removal of the carboxyl group, so what is left is an amine. left is an amine. So glycine participates in synthesis of dash heme, using production of heme. Heme is what is found in hemoglobin. So protein folding is driven by non-polar residues involving, when the answer is already in the question. The answer is already in the question. There's a protein folding is driven by dash, is driven by non-polar residues involves. So protein folding driven by non-polar residues involve dash. The non-polar residue is already telling you that it is hydrophobic. So anything that is non-polar is hydrophobic. So non-polar residue involves in hydrophobic interaction. So the answer to that question is C, hydrophobic interaction. Not hydrogen bonding, not ionic interaction, not ionic attraction, not covalent bonding, or hydrophobic interaction. So do not forget to subscribe and click on the notification bell. So in our next video, we'll be going into same amino acid in our lecture series. Until we conclude amino acid, we're gonna go into lipids and carbohydrates.